Imagine for a second that you are shrinking.
Okay.
You know the vibe, classic sci-fi. Think Fantastic Voyage from the 60s, or maybe Inner Space if you grew up in the 80s like me.
Right.
You're in this tiny, microscopic submarine, shrinking down past the size of a coin, past a grain of sand, until you are navigating the rapids of the bloodstream.
And hopefully without Dennis Quaid piloting the ship, if we can help it.
Ideally, yes. But usually when we tell that story, we're staring out the window at all the biological stuff.
Mm-hmm.
The red blood cells bouncing off the windshield, the white blood cells hunting down bacteria. It's a messy, squishy, chaotic world down there.
It is. Biology is wet, it's dynamic, and it's incredibly flexible. That's, the hallmark of life. It moves.
Exactly. But today, we aren't talking about the biological stuff we're used to seeing. We are talking about the submarine itself.
Okay.
We're talking about building a vehicle or a container out of the very code of life itself.
Well,
We are used to thinking of DNA as a blueprint, right?
the instruction manual.
The instruction manual. But what if DNA wasn't just the instructions? What if DNA was the brick?
And not just a rigid brick, like a Lego block, which is, frankly, how we've treated DNA nanotechnology for a long, long time.
Right.
But a flexible, moving, squishy building material.
Mm-hmm.
Something that can build a structure as complex and dynamic as a living cell.
That is the mind-bending twist we are unpacking today. We are welcoming you to the Deep Dive Lab, and we are looking at a brand new study just published in Nature Materials.
This just dropped.
Yeah, late 2025. It comes from a team led by Christoph Karfuser and Friedrich C. Simmel at the Technical University of Munich, and they have created something called Dipids.
Dipids. It's a catchy name.
It is.
It sounds a bit like a snack food you'd buy at the movie theater.
It really does. Pass the Dipids. But it's actually a clever mashup of words, right?
It is. It's a portmanteau of DNA and lipids. And that name, it basically tells you the whole strategy right there. They're using DNA to mimic the physics of lipids.
Which are the fats that make up the membranes of every cell in your body. So before we get into the how, I want to push back a little on the why. We've had DNA nanotechnology for a while now. We have DNA origami. We've all seen those pictures of the tiny smiley faces in boxes made of folded DNA.
Sure.
Why wasn't that enough? Why do we need to make DNA act like fat?
It all comes down to architecture. If you look at nature, it basically has two architectural styles for building things.
Good.
You have the small and rigid style, and then you have the big and squishy style.
Big and squishy is definitely my post-holiday architectural style. But break that down for me scientifically.
Okay, think of a virus. A virus shell is usually this geometric marvel. It's like a geodesic dome.
Right.
It's made of proteins that lock together perfectly. It's stiff. It protects the cargo inside, but it's a cage. Once it's built, it doesn't really change shape.
It's a locked box.
It is. And for the last 20 years, that's what DNA origami has been. Scientists have gotten incredibly good at folding DNA into these tiny, rigid shapes.
Nanoscale Lego bricks.
Exactly. You click them together, they stay put.
Which is great if you're building a statue, but not so great if you're trying to build a life form.
Precisely. A human cell, any living cell really, is basically a soap bubble. It's a fluid membrane made of lipids. It can stretch, it can split, it can even heal itself if you poke it.
Ah.
If you want to build something that acts like a cell, you can't use a cage. You need a membrane.
And that's been the bottleneck this whole time. We can make the code, but we couldn't make the skin.
We've been trying to build lifelike systems using rigid bricks. And it just doesn't work. You need squishy bricks. And that is exactly what Karfuser, Simmel, and their team at TUM figured out how to make.
They engineered a DNA structure that acts like a lipid.
You got it.
So let's look at this dipid. If I could shrink down to the nanoscale and hold one of these in my hand, what am I looking at? Because when I think DNA, I just picture that long, twisting ladder.
You're looking at a tiny barrel, a cylinder made of folded DNA, about 30 nanometers wide.
Wait, a barrel sounds pretty rigid. I thought we were getting away from rigid.
The barrel is rigid. That's the trick. The individual unit is solid, but the way they connect is totally fluid.
Okay.
Imagine a barrel that's covered in Velcro, but not just little patches of Velcro, long, waving strings of Velcro sticking out everywhere.
Hairy Velcro barrels. Got it. That is a visualization I will not forget.
It's the best way to picture it.
Yeah.
They attached these single-stranded DNA extensions to the sides of the barrels. In the paper, they call them flex domains.
And because they are flexible strings, not like locking mechanisms.
The barrels can move around. When you mix a bunch of them together, those sticky strings grab onto their neighbors, but they don't lock them into a grid.
Ah.
They hold them together loosely.
Hmm.
It creates a sheet that behaves more like like chainmail.
Chainmail is a great analogy. It's made of hard steel rings, but the fabric itself just flows. You can drape it over things.
Exactly.
Yeah.
And this allows the structure to do something that rigid DNA origami never ever could. It can curve, it can bend, and it can close up on itself naturally.
Okay, now this brings us to the assembly process. And this is the part of the paper where I had to stop and reread because it seemed almost too simple. They aren't building these one by one with a microscopic pair of tweezers, are they?
No, that would take 1 billion years. This is all self-assembly. It's all based on geometry.
So how do you tell a bunch of floating barrels to turn themselves into a ball?
You don't tell them. You shape them. The researchers can tune the length of those flexible strings.
Right.
So by making the strings at one end a little tighter than at the other end, they effectively turn the cylinder into a cone.
Like a traffic cone.
A very, very subtle traffic cone.
Yeah.
But think about what happens if you try to pack 100 traffic cones together, side by side on the floor.
Well, because the base is wide and the top is narrow, they can't sit flat. They'd naturally start to curl up.
Bingo. The geometry forces them to curl.
Yeah.
If you pack enough of them, they curl all the way around and close into a sphere. The researchers just program that cone angle into the DNA sequence, throw the bricks in a test tube, and the laws of physics do the rest.
That is wild. You program the brick and the brick just knows how to build the house.
That's the dream of bottom-up engineering. And the result isn't just a tiny little dot. This is where the paper really shocked me. The scale of it.
Yeah, let's talk size, because usually nano means, well, nano, tiny, virus size.
Right. But these Dipids formed containers that were massive. I mean, relatively. They range from what they called extra small, about 100 nanometers, all the way up to XXL, which is over a micron.
Okay, for those of us who don't measure our groceries in microns, give us a comparison.
A micron is the size of an E. coli bacterium.
Whoa.
In the world of DNA nanotechnology, that is absolutely gigantic. We're talking about a structure with a mass of 40 gigadaltons.
I love the word gigadalton. It sounds like a Marvel villain weapon. Beware the gigadalton.
It does. It's a unit of molecular mass. A typical protein might be, say, 50 kilodaltons. This is nearly 1 million times more massive.
So they've built a skyscraper using toothpicks.
Pretty much.
But here's the thing that struck me in the images they included in the paper. These skyscrapers, they aren't perfect. They look a bit lumpy.
They look like deflated soccer balls. Or lopsided balloons, yeah.
And usually in science, lopsided means we messed up. But the researchers seem pretty happy about it.
Oh, they were thrilled. Because that's what real cells look like.
Ah.
Real biological membranes are floppy, they deform, they fluctuate just from thermal energy. If they had come out looking like perfect, rigid spheres, it would have been a failure. The floppiness proves they successfully mimicked the physics of lipids.
So the imperfection is actually the proof of concept.
Exactly.
Okay, so we have a giant, well, relatively giant flexible self-assembling container. But just having a bag doesn't make it a cell. A Ziploc bag is a container, but it's not alive.
Right.
A cell has to be organized. It's not just a soup inside.
This is where the Dipid concept gets really sophisticated. They didn't just make one type of brick. They made type A bricks and type B bricks.
And I'm guessing type A doesn't like to hang out with type B.
They tolerate each other, but they prefer their own kind. It's like oil and water. So if you mix them together to form a sphere, they separate into patches.
Okay.
You get a sphere that is mostly type A, with a big island or a raft of type B floating in it.
This is that patchy particle concept from the study. But why do we care about patches? Why does it matter if my artificial cell has spots?
Because that's how you program functionality. Imagine you want this artificial cell to stick to a tumor.
Right.
You don't want the whole thing to be sticky. It would get caught on everything in your blood vessels. You want one specific spot to be sticky.
Ah, okay. So you program the type B patch to have a Velcro hook for cancer cells, and the rest of the cell is smooth so it can slide through the body.
Exactly. You can spatially organize the surface. You can put the engine on the back and the sensors on the front. This is incredibly hard to do with regular soap bubbles, which are just random mixtures.
But with DNA.
With DNA, every single pixel on that surface is addressable.
That is the power of using DNA, isn't it? It's programmable matter.
It is. And the team at TU Munich proved they could control this layout with incredible precision.
So we have the structure, we have the layout, but a cell is a machine. It does things. It, breathes, it eats, it makes proteins. If this is just a sealed plastic bag, it's useless.
It needs to interact with the world.
Yeah.
We're talking about permeability. And this is the hardest tightrope walk in cellular engineering. A cell needs to be a fortress that keeps its DNA safe inside. But it also needs to be a marketplace that lets food in and waste out.
Semi-permeable.
Right. Now, how do you make a wall made of barrels semi-permeable?
I guess. The barrels themselves are hollow.
The barrels are hollow, yes, but also the chainmail weave has gaps in it. The researchers found that the membrane is naturally porous.
Okay.
Small molecules, sugar, water, tiny fuel sources, they can drift right through the walls. But big molecules like enzymes, they're trapped.
So it's a filter. It keeps the furniture inside the house, but lets the air flow through the windows.
It's a selective barrier. And they proved this with an experiment that I think is just the coolest visual in the whole paper, the glowing green experiment.
Yes. This involves broccoli. Please tell me they put actual broccoli inside a DNA robot.
I wish. No, it's broccoli.
Ah.
It's sadly not a vegetable. It's an aptamer.
We need a quick sidebar here. What is an aptamer?
Think of it like a chemical light switch. It's a piece of genetic code that is folded in a specific way so that when it grabs onto a certain dye molecule, it glows bright green.
Okay, a light switch.
But here's the catch. It only works if it's transcribed into RNA. It needs to be activated by a machine.
So they put the code for this light switch inside the artificial cell.
They pasted the DNA template to the inside wall of the Dipid container. Then they put the enzyme, the machine that reads the DNA, called RNA polymerase, on the outside.
So the machine is outside, the blueprint is inside.
Right. They wanted to see if the machine could get in. And sure enough, because the membrane is porous, the enzyme swam through the gaps in the chainmail.
No way.
Found the template inside and started churning out RNA. And suddenly the inside of these artificial cells started glowing green.
That's profound. It proves the thing isn't just a dead container, it's a reaction chamber.
It proves it can host biochemistry. It can protect a reaction from the outside world while still drawing fuel from it. That is essentially the definition of metabolism.
And they didn't stop at one reaction. They went full inception.
The Matryoshka doll effect. They managed to trap a small Dipid container inside a giant Dipid container.
Why? Just to show off. Look, we put a ship in a bottle.
No, this is absolutely key to advanced life. Look at your own cells. You aren't just a bag of fluid. You have a nucleus. You have mitochondria.
Organelles. Little rooms inside the big house.
Little rooms. And each room does a different job.
Compartmentalization.
Exactly. If you want to build a complex artificial cell, you need rooms. You need one room to generate energy, another to break down toxins, another to store data. By putting a container inside a container, the TUM team showed that Dipids can support this kind of hierarchy.
It feels like we are inching incredibly close to something that just blurs the line between material and creature.
We really are. The authors mentioned soft robotics in the paper, but I think even that term is too mechanical.
When I hear soft robot, I think of that squishy Baymax character from the movies.
It's a similar idea. But on a microscopic scale, imagine a Dickid robot in your bloodstream. It's flexible, so it squeezes through capillaries without clogging them.
And it has that patchy surface we talked about.
So it only targets specific cells.
Like an artificial immune cell?
Yes. It could swim up to a virus, open a pore, swallow it, and digest it inside a sealed chamber using enzymes. Or it could deliver a drug directly into a tumor cell. It's wetware.
Wetware. That's the word. It's programmable medicine. But here's the question that sticks with me, and I want your honest take on this.
Okay.
We have the shell. We have the compartments. We have the ability to let fuel in. What's missing? Why isn't this life?
That's the big philosophical hurdle. Currently, these things self-assemble, but they don't reproduce. They don't copy themselves.
They don't divide.
Not yet, but the paper hints at it. Because these are modular, they're just bricks floating in a soup.
Yeah.
You could theoretically create a system where the membrane grows, gets too big, and then pinches off into two daughter cells.
That's the next horizon.
That's the holy grail.
Yeah.
If they can get these Dipids to grow and divide, then the line between synthetic biology and actual biology basically vanishes.
It's interesting to think about the tool set. For decades, biology was something we just studied. Right. It was observation. We looked through the microscope and we took notes. Now it feels like biology is becoming a manufacturing discipline.
It's moving from reading nature to writing nature. And what I love about this specific paper is that it admits nature was right all along.
How so?
We spent decades trying to force rigid DNA geometry onto a biological world. It didn't work. It was only when we embraced this squishy, chaotic, flexible physics of nature that we finally built something at the cell scale.
We had to stop fighting the chaos and start engineering with it.
Geometry is destiny, but only if you use the right materials.
I think about the applications here, and it's just dizzying. We've talked about medicine, but could this be used for other things?
Oh, absolutely. Think about sensors. You could have these Dipids floating in a water supply detecting toxins,
And if they detect a pollutant, they glow green.
just like the deep broccoli experiment. Or think about chemical manufacturing. Instead of these huge factories, you have vats of artificial cells just churning out complex medicines or fuels.
It's the ultimate efficient factory.
And because it's made of DNA, it's biodegradable. It's not plastic, it's biological material.
So what's the takeaway here for the person listening who might be stuck in traffic or doing the dishes? We aren't going to see these in the pharmacy tomorrow.
No. But this is a platform. That's the key word. Carfuse and Simmel haven't just built a new gadget, they've created a whole new construction system.
A new box of Lego.
They've given the scientific community a new box of Legos, squishy Legos, and said, go build something.
We're going to see a lot of weird, wonderful, and probably slightly terrifying things built out of these Dipids in the next decade.
I fully expect to see artificial organelles, maybe even artificial photosynthesis systems built using this framework. We are entering the era of squishyware,
Well, I, for one, welcome our new squishy DNA overlords.
as long as they're friendly.
Huge thanks to the team at TU Munich for this mind-bending research, and thank you for helping us unpack it. It's always a trip to see just how advanced this field is getting.
My pleasure. The micro world is getting more interesting by the day.
And thanks you for listening to the Deep Dive Lab. If you enjoyed this trip into the nanoscale and you want to keep up with the future of wetware, go ahead and hit that subscribe button. And hey, I want to leave you with a thought.
Go for it.
If we can build the body of a cell, and we can put the engine of a cell inside it, at what point do we stop calling it a machine and start calling it a creature?
That is the question of the century.
Mull on that. We'll see you in the next deep dive.